What if I told you that a virus isn't just a pathogen, but a master code breaker, a tiny intelligence operative pulling off a sophisticated heist right at the molecular level? Well, today, we're cracking open the secret playbook of plant viruses. And believe me, what's inside is an absolute masterclass in molecular deception. So, picture this. Every single plant cell is like its own high-security factory. It is humming with activity, and it follows a super strict set of rules, kind of like an operating system, to produce all the proteins it needs to live. It's a fortress, and it's built to keep any unauthorized programs out. And this is the big mystery, isn't it? How does this tiny, seemingly simple thing, which is really just a bit of genetic code wrapped in a protein coat, how does it possibly breach those defenses and hijack the entire factory for its own ends? Well, the answer is it doesn't use brute force. It uses espionage. It has this incredible playbook of tricks to fool the cell's machinery, to bend the rules, and to stage a complete and total corporate takeover. And we're about to look at some of its most mind-blowing tactics. Okay, so first things first, to really appreciate the genius of this heist, we have to understand the security of the vault. So let's take a closer look at the fortress the virus needs to crack, the cell's protein production line. At the heart of this factory is a process called translation. You can think of it as a hyper-secure operating system. It takes a genetic blueprint, which we call messenger RNA or mRNA, and it reads it to build a very specific protein. But, and this is the key, it is incredibly rigid. It only reads blueprints that are formatted in a very, very specific way. In fact, you can think of the system as having 3 ironclad security protocols that any incoming message has to follow, no exceptions. These protocols are what make life possible, and they're what make a virus's job seem almost impossible. So, these protocols create what is, for our virus, a nearly impossible mission. We're talking a microscopic mission impossible, and these protocols are the laser grids it has to somehow navigate. So what are these hurdles? Well, viruses face 3 huge problems. First, the polycistronic problem. The cell's machinery reads one blueprint, makes one protein, and then it's done. But a viral blueprint needs to code for lots of different proteins. Big problem. Second, the cap accessibility problem. Every official blueprint from the cell has a special security badge on the front, called a 5-cap. No badge, no entry. The cell's machinery won't even look at it. And guess what? Most viruses don't have one. And third, there's the replication-translation conflict. The factory reads blueprints from front to back. But for the virus to make copies of itself, it needs to copy its blueprint from back to front. It's a head-on collision just waiting to happen. Seems like game over, right? So how do they solve that first problem, that one blueprint, one product rule? Well, some viruses have evolved a truly radical solution. They just shatter their blueprint into pieces. This is called the multipartite strategy. Now, a standard virus tries to sneak in one long, illegal blueprint that has instructions for everything. It's kind of risky. But a multipartite virus, it breaks its entire genome into multiple, separate pieces. Each piece has just 1 or 2 genes, so each one looks like a small, legitimate, one-product blueprint that the cell's machinery is totally happy to read. So clever. And get this isn't just about sneaking past security. It gives the virus an incredible advantage. It can control the dosage of its proteins. It doesn't need the same amount of everything, right? It needs tons of coat proteins to build new virus particles, but only a few of the replication enzymes. So it just makes way more copies of the blueprint pieces it needs most. This is known as the genome formula. Just take a look at this. A virus like the fava bean necrotic stunt virus can maintain a really high number of blueprint segments for its coat protein, flooding the factory with those while keeping the number of segments for its replicase enzyme really low. I mean, we're talking about regulating production by up to 100-fold, and it's hard-coded right into the genome structure. That is unbelievably efficient. Okay, so shattering your genome is one way to go. But what if you can't do that? Well, then you have to use an even more subtle tactic. Some viruses don't just bypass the rules. They actively rewrite them using coded deceptions. This is where it gets really, really cool. These viruses, they let the cell's machinery, the ribosome, start reading the blueprint just like normal. But then, at a very specific, pre-programmed spot, they use a hidden signal in the RNA code to trick that ribosome into making a mistake, like a tiny molecular stutter or a hiccup. Now, this recoding usually comes in two main flavors. The first is called stop codon read-through, which is basically a leaky stop sign. The ribosome is supposed to stop, but it gets tricked into just blowing right past it, making a much longer protein. The second one, though, is even more mind-bending. It's like a secret trapdoor that shifts the ribosome's entire reading frame. This is programmed ribosomal frame shifting, and it is a beautiful two-part trap. First, the virus lays down some bait, a specific 7-letter sequence of RNA, like GGG-UUU, that's designed to be slippery and hard for the ribosomes to read, causing it to pause. Then comes the trap. Just a little bit further down, the RNA is tied into this complex structural knot called a pseudoknot. So when the paused ribosome tries to move forward, it bumps into this knot, creating tension. And that tension forces the ribosome to slip backward by exactly 1 letter on that slippery sequence. Now it's reading a completely different set of instructions, producing a totally new protein from the same blueprint. Amazing. And in some viruses, it's even more complex than that. This whole frame shift mechanism, this trapdoor, is controlled by a master switch, a molecular bridge formed by the RNA folding back on itself, connecting the trapdoor to a point nearly 4,000 letters away at the other end of the genome. This isn't just a simple trick. This is long-distance, structural engineering that coordinates the entire viral life cycle. It's a level of nanoscale control that scientists are still dreaming of being able to replicate. Okay, so we've seen how viruses solve the one blueprint problem and literally rewrite the rules of translation. But what about that security badge, the 5' cap? Without it, the factory's guards aren't supposed to let you in. Well, if you don't have a key, you can always pick the lock. Or, in the virus's case, you just forge a key that looks exactly like the real thing. Viruses have come up with these unbelievable ways to mimic the function of that 5' cap. And there are two main forgery strategies. On the left, we've got the RNA key. Some viruses have this complex, folded-up piece of RNA at the back end of their genome. This structure acts like a powerful magnet for the cell's machinery. Then the RNA strand literally folds over on itself, and two specific loops of code touch each other, and this contact acts like a teleportation device, delivering the machinery right to the starting line at the front. And on the right, you have the protein key. This is even more direct. Viruses like potivirus just stick one of their own proteins, called VPG, right onto the front of their RNA. And this VPG protein is a perfect molecular mimic of the real cap, fooling one of the cell's main security guards completely. So we've seen shattered blueprints, coded deceptions, forged keys. These aren't just fascinating biological quirks. Understanding this playbook is a matter of global security. This is a molecular arms race, and the stakes are incredibly high. So let's just do a quick recap of the heist. The shattered blueprint, the multipartite genome, that's all about controlling protein dosage. The coded deception of the ribosomal frame shift, that's a master switch between making proteins and making more copies of the virus. And the forged key, whether it's an RNA structure or a VPG protein, that's all about hijacking the host's machinery from the get-go. And this is why it all matters. Every one of these tactics is a potential vulnerability. If we understand exactly how that protein key fits into the cellular lock, we can breed plants with a slightly different lock that the key no longer fits. If we know the exact structure of that frame-shifting knot, we can design molecules to jam it up. This isn't just basic science. This is the blueprint for protecting our global food supply. You know, this back and forth between plants and viruses is one of the oldest wars on our planet. And as our climate changes, it puts new stresses on plants, which can change the cellular battlefield and give viruses new openings. So the question that we're left with is, in this ancient and ongoing molecular war, who's going to evolve the next masterstroke? The cellular fortress or the viral codebreaker?